Hi, today I'm here to discuss my project of developing a new supersensitive mesoscale discovery biomarker immunoassay for chronic kidney and eye disease. A little bit of background. Diabetes is the #1 cause of new cases of kidney failure, with the number of people with diabetes rising from 108 million in 1980 to 422 million in 2014, according to a report by the World Health Organization. Between the years of 2000 and 2019, there was a 3% increase in diabetes mortality rates by age, while in 2019, diabetic kidney disease caused an estimated 2 million deaths, and people with diabetes are 6 to 12 times more likely to be hospitalized with chronic kidney disease or kidney failure. At the same time, a study concluded that an estimated 9.6 million people in the United States suffer from diabetic retinopathy, an eye condition which could eventually affect vision or lead to blindness in diabetic patients. Published studies have indicated that there is a strong association between chronic kidney disease and diabetic retinopathy, with both having common risk factors such as genetic and cardiometabolic factors, oxidative stress, inflammation, endothelial dysfunction, microvascular dysfunction, atherosclerosis, among other factors, and both have similar pathogenic mechanisms. Furthermore, as seen in the graphic here, there are similar structures that exist in the kidney in the form of the blood-urine barrier and in the eye with the blood-retina barrier. Around the glomerular basement membrane and the Brooks membrane in the eye show certain similarities, which further emphasizes the potential for studies to extend over between the two structures. Published studies indicate that SLIT is a potential biomarker for gestational diabetes. SLIT is a protein ligand that plays an important role in the SLIT-ROBL signaling pathway that has a significant impact on podocyte adhesion in the kidney, which could affect protein leaking into the urine or the bloodstream due to changes in the glomerular basement membrane, and is something that we have been studying for a while. SLIT expression has also been found to be upregulated in vitreous fluid samples from patients with diabetic retinopathy. As we know, diabetes can cause both diabetic retinopathy and diabetic nephropathy, or diabetic kidney disease. However, the gold standard of the regular commercially available SLIT ELISA assay, as used in this study, has a very limited dynamic range with low quantification detection limits for SLIT. Additionally, the data from this study with samples of proliferative diabetic retinopathy lying around 1 nanogram per milliliter for 1,000 picograms per milliliter lies right around that low limit of detection as seen next to the graph, where the lower level of detection is only 1,000 picograms per milliliter. As seen in the error bars, this leads to a wide range of data points and renders the data unreliable and unreproducible. Our objective is to create an immunoassay that can accurately and reliably quantify low levels of SLIT protein in eye vitreous fluid samples, and then use that to try to determine whether SLIT can reliably act as a biomarker for diabetic retinopathy, and potentially, by extension, diabetic nephropathy. We used a new MSD immunoassay technology, which utilizes electrochemiluminescence to measure and quantify our low protein levels in samples. Comparing the new MSD technology to a traditional ELISA assay, it requires about 1/4 of the sample levels and provides a more sensitive and precise result. And it handles complex biological matrices more easily and uses simpler, faster protocols. It can also quantify up to 10 analytes per well, and it measures elevated and normal expression in samples without the need for multiple dilutions, as we would need in a traditional ELISA. These advantages that the MSD technology provides us allows us to better measure and analyze wider ranges of data and to better understand the relationship between SLT protein and diabetic eye and kidney diseases. Similar to the typical ELISA assay, the MSD immunoassay sandwiches the SLT protein between a capture antibody anchored to a 96-well plate through strip avidin conjugation and a detection antibody. However, instead of using enzyme reactions to produce luminescence and quantify it, the new MSD technology allows us to run an electrical current through the samples, which would excite a ruthenium ion, which had been conjugated onto the detection antibody, allowing us to measure the level of excitation and provide reliable and accurate quantification of low protein levels. First, we use various concentrations of antibody to try to optimize our assay for future use, and to also ensure that our new assay provides a greater range of detection and one that is more precise and more suitable for our low protein levels. As seen in the graph, the expected levels of around 1,000 picograms per milliliter lies perfectly within our detection range, and the range extends all the way to a lower limit of about 5 picograms per milliliter, which is significantly lower than the standard ELISA assay. Additionally, we have found that the vitreous eye fluid SLT protein levels to be 7-fold higher than that in serum samples. This is contrary to previous studies that have indicated that SLT levels would be 10-fold higher in serum samples, and it further shows the unreliability of commercially available ELISA assays. After running statistical tests on our data, we have found upregulation of SLT in diabetic retinopathy samples, as seen in our pilot study in this graphic. However, due to the low number of samples, we cannot make a full conclusion based on the existing data, and further experimentation must be done to confirm our findings. At the moment, though, we can see that there is still a trend that indicates SLT could potentially be a biomarker for diabetic retinopathy through vitreous fluid in the eye. In summary, we have developed a highly sensitive SLT MSD biomarker immunoassay, and we optimize concentrations of antibody to more efficiently run our assays. We also found SLT concentration in vitreous fluid to be 7 times higher than that in serum, and our pilot study indicates that there is a trend of increased SLT concentration in vitreous fluid samples in diabetic retinopathy patients compared to non-diabetic patients. However, additional vitreous samples are necessary in order to confirm our findings. Special thanks to Dr. Liu, Dr. Kumar, Dr. Fan, and my peers in the Liu lab, as well as Dr. Weiker's lab and Dr. Subramanian's lab in ophthalmology, as well as MSD for their help in creating this assay. Furthermore, I'd like to thank the people who fund this project. Thank you.